Okay, ladies and gentlemen, good day all. My name is Mukhtar Ali from Department of Biochemistry, ABU Zadia. Today, we are going to look at the concept of enzymes catalysis. Please, if you are coming for the first time to my YouTube channel, please subscribe. What is enzymes? Enzymes are catalysts. They increase the speed of chemical reaction without themselves undergoing any permanent chemical change. So it means that enzymes, they are molecules, or they are proteinous in nature, or they are protein molecules that speed or that increase the chemical reactions in a cell. Generally, there are thousands of reactions that are taking place in our body, and some reactions can take years before they complete. So, as a result of that, if there is a slow in biochemical reactions in a cell, so there is a likelihood that the energy or the demand of the cells cannot be the demand of the cells cannot be met. So as a result of that, there are enzymes which generally speed up the reactions. Instead of the reactions to take years, instead of the reactions to take months, instead of the reactions to take hours, in just milliseconds, they will easily convert that reactant to products. So generally, when we are talking about reactions, we're talking about reactants and products. But in the case of enzymes, we are not usually called reactant as a reactant. Rather, we call them substrates. So generally, enzymes, they are molecules that speed the rate of the reaction so that the reactions cannot take years, cannot take hours, because as long as the reactions is not taking place at a required time, so there's a likelihood that the cells will die. And of course, when the cells is die, it means that the living organisms will entirely die. So that is why these enzymes are very, very important. They are very, very essential in living organisms. So they increase the rate of the reactions without themselves being consumed. So they do not change. If enzymes A combine substrate A to product A or to product P, means that the enzymes will retain its integrity, it will retain its chemical nature, it will not change, and it is recycled and used to combat another substrate. So that is in the case of enzymes. So enzymes, they are biological molecules that speed the rate of biochemical reactions, and they do not consume. Nothing is changed on the enzymes, but they change the substrate to product. So enzymes neither speed off in the reaction, nor they appear as reaction product. So they do not use off, and they do not appear as reaction products. So they are not used off, and you cannot see them in the product produced. So it means that what the enzymes do is just, it will change the substrate, the product, without it being used off and without appearing on the product produced. So these are some characteristics of enzymes. Generally, enzymes are high molecular weight proteins, although it's not all enzymes that are proteins. There are some enzymes that are nucleic acid in nature, but 99.99% of the enzymes are proteinous, but very few are nucleic acid in nature. So they have high molecular weights, which range from 10,000 to 2 million grams per mole. So it means that their molecular weight is huge. And the substrates, specific catalysts, so they are specific, recognize and catalyze only their substrates. If enzymes is responsible for converting glucose to glucose 6-phosphate, that the same enzymes cannot be used to convert fructose 2 fructose 6-phosphate. So it's not possible. So enzymes, they are specific, they are substrates, and they have high efficiency. They are highly efficient. They are highly efficient, increasing reaction rate by a factor as high as 10 power 8. So it means that enzymes, they increase the rate of the reaction by a factor of 10 power 8. Is huge. So the factor is huge. So that is why enzymes reaction is very, very essential, as it's reduced the time for the reaction to take place. So it increases the reaction by a factor of 10 power 8. And then we have enzymes nomenclature. So the earliest enzymes that were discovered that was discovered have become names. Like for example, in the olden days, pepsin, renin, trypsin, pancreatin, that is the name of most of the enzymes. But with some development, with an IFAC achievement, the enzymes named for most other enzymes end in ASE. So that is why now you consider most of the enzymes, their name is ending with ASE. Kinase, amylase, lipase, sucrase. So most of the enzymes, anywhere you see the word end with ASE, it means that that substance is an enzyme, or that molecules is an enzyme. So they usually end with ASE. So the enzyme's name, usually the enzyme name indicates the substrate acted opened and the type of reaction that it catalyzes. So in most cases, if you know, or if you see enzymes, you can clearly see them with the names of the substrate that they act on. Like we have an enzyme called glucokinase because they act on glucose. We have some enzymes called hexokinase because they act on 6-carbon sugar. We have some enzymes called glyceraldehyde 3-phosphate dehydrogenase because they act on the hydrogen. It act on glyceraldehyde 3-phosphate. So most of the enzymes, they have the name of the substrate they act on and the types of reaction that it catalyzes. So like for example, glyceraldehyde 3-phosphate dehydrogenase, it means it's an enzyme that catalyzes dehydrogenation of glyceraldehyde 3-phosphate. So most of the enzymes, they have the name of their substrate that they act on and also the types of the reaction that they catalyze. We have some transferase enzymes like alanine aminotransferase. So most of the enzymes have the name of the substrate and the types of the reaction that they catalyze. So enzymes name, examples of enzymes name, we have glutamic oxaloacetic transaminase. You see this one from the word transaminase, it means it catalyzes transamination, that is the transfer of amino group. And of course, it acts on the substrate is glutamic acid. So we also have L-aspartate 2 oxaloglutarate aminotransferase. So the enzymes name tends to be long and complicated. They are often abbreviated with acronyms. So in most cases. So how these enzymes catalyze reaction? So one of the mechanisms of the enzymes catalysis is the enzymes usually have active sites where the substrates bind to. So this is a substrate. This is our substrate. So the substrate here will now bind to the active site of the enzyme. So this is the active site of these enzymes. So it will now bind to the active site. So the enzymes and the substrates react and form what we call enzyme-substrate complex. And the enzyme-substrate complex will now break down to produce the free enzymes and the substrates. So now if you look at it from the substrates, the product, there is a change in substrate to product in terms of color and the number of products produced. So it means that from one single substrate, two products are generated. So from one substrate, two products are generated and the color has changed. But in the case of enzymes, what you have before the reaction and after the reaction is the same. So that means that the enzymes are not used off and they do not change in any form. So that is the enzymes catalysis. So the enzymes usually bind to the substrates using the active sites. The enzyme substrate complex will be generated, and then finally the enzymes will form a product. So the product usually change from substrates, and then the enzymes doesn't change in the course of the chemical reaction. So that is the mechanisms of how enzymes catalyze substrates. So how is this enzymes catalyze the rate of biochemical reaction? So the way enzymes catalyze biochemical reaction is actually through reducing the energy barrier, which is activation energy. So this is an uncatalyzed reaction. So generally for a chemical reactions to occur, the reactant particles must produce or must collide with one another, and the collision must be perfectly elastic so that the substrate will now produce a sufficient energy to overcome the activation energy before the reaction takes place. So before the reactant produce a product, the reactant must collide to produce sufficient energy that will overcome the energy barrier, which is the activation energy. So in the case of enzymes catalyzed reaction, what the enzymes do, it's just reduce the activation energy so that the substrate can easily react to produce a product. And that is how the enzyme speeds the rate of the chemical reaction, is through reducing the activation energy, that is the energy barrier. And of course, if you can see it clearly here, you see that the activation energy here is high, while the activation energy EA here is lower compared to the uncatalyzed reaction. So that is why this substrate, for this substrate to easily convert to produce a product here, is simple compared to from here to here. So it means that in the absence of enzymes, there must be a sufficient energy that will now jump or overcome the activation energy. But here, when there is an enzyme, the enzyme just reduces the activation energy so that the substrate can easily convert to product. So that is how enzymes increase the rate of the reaction, is by reducing or decreasing the activation energy of the reaction. So now we are now going to look at the enzyme specificity. So the action of an enzyme depends primarily on tertiary and quaternary structure of the protein that constitutes the enzymes. And of course, in the case of enzymes, we have enzymes are proteins, and of course proteins have various levels of structure. We have primary structure, we have secondary structure, we have tertiary structure and quaternary structure. So most of the enzymes are in tertiary and quaternary structure, and it's actually the tertiary and quaternary structure that make them to carry out their enzymes catalysis. But once their tertiary and quaternary structure is being destroyed, it means that the enzyme's activity itself will also be destroyed. So the action of an enzyme is primarily depend on the tertiary and quaternary structure of the protein. So it means that if the tertiary and quaternary structure of the enzyme's proteins is destroyed, it's denatured, it means that there will be no activity. And some of the things that destroy the active site of, or that destroy the tertiary and quaternary structure of the proteins include high temperature, high ionic strengths, and so many other factors like pH. So, and the part of the enzyme structure that acts on the substrate is the active sites. So there is a fat that is found on the enzymes called active sites, where the substrate binds for the catalysis to take place. And in this active site of the enzymes, there are amino acids. There are amino acid which is called amino acid residues. So it means that in the active sites in the enzymes, there is a cleft or there is a pocket called active site where the substrate binds for the catalysis to take place. So this active site of the enzymes, there are amino acids that are found which are involved in the catalysis. And these amino acids that are found in the active site of an enzymes are called amino acid residues. They are called amino acid residues. And usually in the active site of the enzymes, the amino acids are usually far apart. They are far apart. They are lengthy. They are far apart. They are not in close proximity. So the active sites of the enzymes is a groove or a pocket in the enzyme structure where the substrate combines. So this is specifically, if you can now see, this is an enzymes E, and of course this is the active site. So everything here is called active site. Everything here is called active site of the enzymes. So this is the active site where the substrate, and this is the substrate where the substrate binds. So it's called, this is the enzymes entirely, and this is the pockets called active site that are found in the enzymes. And this active site of the enzymes is divided into two. We have binding site and we have catalytic site. So the active site of the enzymes is divided into two, which is called binding site and catalytic site. So what is the difference between binding site and catalytic site of an enzyme? The binding site of the enzyme, it usually chooses the substrate and binds it to the active sites. Of course, the enzymes are very specific. The binding site of the enzymes is the site where usually chooses the perfect substrates or the specific substrates and binds to it. And that is what makes enzymes to be specific. Like for example, if you have glucose and fructose and the enzymes is meant to catalyze, let's say the enzymes is glucokinase and is meant only to catalyze glucose, not substrate. So it means that the binding site of the enzyme is the site that has the responsibility of choosing glucose, not fructose, and then bind it to the active site. While after binding it to the active site, then the catalytic site, it is the one that performs the catalytic action of the enzymes. So the binding site is the site that recognizes the substrate and binds to the active site, and the active site will now undergo the catalytic activity of the substrate to products. So this is the case of enzyme specificity. So enzymes are specific because of the binding site that usually recognize a required or a specific substrate, bind to it, and allow the catalytic site to carry its enzymatic activity or the catalytic activity. So that is for the enzymes activity. And then the next thing is cofactors. Cofactors. What are cofactors? Cofactors are other compounds or ions that enzymes require before their catalytic activity can occur. So cofactors, they are compounds, they are other compounds, and they are not proteins. They are not proteins, but they are compounds or ions that enzymes require before their catalytic activity can occur. It means that if there is no cofactors, there will be no catalytic activity. So the protein portion of an enzyme is referred to as enzymes. So in the enzymes, we have different portion. We have protein portion and we have non-protein portion. So the protein portion of an enzyme is called enzymes. And then the enzymes plus the cofactor, when you have enzymes and you add it with the cofactors, is what you call holoenzymes. So holoenzymes is the active form of the enzymes. So if there is no cofactors, it means you have enzyme. And if it is only enzymes you have, it means that the enzymes is inactive. If it is enzymes, it indicates that the enzymes, alpha enzymes, if you have alpha enzymes, which is the protein portion, is usually inactive. And there is cofactors, and it's these cofactors, when binds to the enzymes, then it now become the whole enzymes, which is called holo enzymes. Holo enzymes means whole enzymes, and it's the active form of the enzyme. So if there is no holo enzymes, if there is no holo enzymes, it means, sorry, if there is no cofactors, it means you only have alpha enzymes, and alpha enzymes cannot work, is inactive. So these cofactors is divided into three. We have essential ions, and we have coenzymes, and we have prosthetic groups. So what is the difference between coenzymes and prosthetic groups? So the coenzymes, they are molecules that are loosely bound to the alpha enzymes. They are not that tightly bound. But we have prosthetic group, which is the one that strongly bound to the alpha enzymes. That is prosthetic group. And then we also have essential ions, and these essential ions can be activator metal ions, which is usually loosely bound. Like, for example, in the case of hexokinases, an enzyme that convert glucose to glucose 6-phosphates, and the enzymes need activator metal. So example of the activator metal of hexokinase is magnesium. So it's loosely bonded. Like, for example, let me just give examples of this, of one reaction. So in a situation where we have like glucose plus ATP, an enzyme is called hexokinase, and need a substrate, and so they need a cofactor, and the cofactor is magnesium ion for it to produce glucose 6-phosphates. So these kinds of activator ions this is a typical example of an activator ions that is loosely bound. But we have metal ions of metalloenzymes, which is tightly bonded. Like, for example, in the case of hemoglobin, we have ion Fe. So it's an example of metal ions of metalloenzymes. It means that they are the enzymes that are, or they are the proteins that they have a metal ions that is from the part of the proteins or enzymes. So that's why they are called metalloenzymes, because they are enzymes that have cofactors or metal ions that fully integrate to it. So that is for that is the cofactors, and we have some examples of the cofactors here. Like, we have iron Fe, we have manganese, we have magnesium, and we have zinc. So these ions that are here are typical examples of the essential ions that are found in proteins or in enzymes. So, ladies and gentlemen, if this is the time you actually visit this is the first time you visit my YouTube channel, please subscribe. Please subscribe for more content. So let's meet in the next video of enzymes. Thank you for your time, and I hope you like the video.